Hello again. If you reached this video, I hope you're enjoying this as much as I am. So the second lipoprotein we're going to talk about is the VLDL. The VLDL is actually a lipoprotein that is synthesized from endogenous fat from the liver. Okay? It's synthesized from the liver. The type of fat it contains is triacylglyceride and very, very low content of cholesterol to the extrahepatic tissue. So we have to know this information. The VLDL gets synthesized from the liver to the extrahepatic tissue. From the liver to the extrahepatic tissue. So the first thing is formation of a nascent VLDL, which is the structural component, which is the B100 also. The formation of the VLDL is very similar to the chylomicron. The HDL is going to donate its ApoC and ApoE to the VLDL. Okay, but the thing is different here is that the VLDL has a much lower triacylglyceride and higher component of cholesterol than the chylomicron. So as I said, the ApoC2 is going to activate the lipoprotein lipase C, and the ApoE is going to mediate or help in the uptake by the liver later on. So formation of the remnant VLDL. The remnant VLDL have another name, which is the IDL, the intermediate density lipoprotein. So it's going to be taken up. So when we have IDL, it's going to be taken up by the liver, by the ApoE proteins. But some of the IDL, some of this intermediate density lipoprotein, is going to become LDL. When it becomes LDL, this is actually the lipoprotein cascade. You have to know this sequence. So first we had the VLDL synthesized by the liver, and later on we have this VLDL becoming a mature VLDL by having the ApoC2 and the ApoE from the HDL. Then we have the remnant VLDL, which is also called the IDL. Some of the IDL is going to become LDL, the low density lipoprotein. And if the triacylglyceride synthesis is very high and we have very low VLDLs, that means the liver is not synthesizing enough VLDLs. It's actually keeping the TGs inside the liver. There's a problem, and it's going to cause fatty liver. So this is also the other disease we have, which is the fatty liver, caused by low amount of VLDL. Because VLDL is the one transporting the triacylglyceride outside the liver, the endogenous fat. And if we had very low amounts of the VLDL, that means the triacylglycerides are inside the liver, and it's going to cause the liver to be fatty. So we said the LDL, the low density lipoprotein, is derived from the IDL, the remnant of the VLDL. So the LDL, which is the third type of lipoprotein we have here, the LDL is actually formed from the VLDL remnant, so that will be uptaken by tissues. How? So let's see. So ApoB100 on the LDL, but inside we only have cholesterol. So the ApoB100 acts as a ligand for LDL receptor. So this LDL is containing cholesterol is going to have 30% going onto the extrahepatic tissue, the vessels, and 70% on the liver. So it's bad because it actually transfers or deposits its cholesterol on the vessels, on the vessel wall. So because we have B100 receptor, and here we have the ApoB100. So this is the LDL, the low density lipoprotein. So if intracellularly we have cholesterol level is high, so how we're going to regulate that? By two mechanisms. First, the suppression of the LDL receptor synthesis by cholesterol. So if we have high cholesterol levels in the blood, the cholesterol is going to have this induced mechanism. It's going to suppress the LDL receptors This is the first mechanism. This will decrease the rate of the LDL removed from the serum. So when this decrease of LDL, we're going to have a lot of cholesterol because it's not removed by the receptors. So we're going to have atherosclerosis, leading to increase in the cholesterol in the blood because suppression of these LDL receptors. So we don't have enough receptors to recognize these cholesterol and move it elsewhere. It's going to be stuck there in the blood, cholesterol in the blood, causing atherosclerosis. So this is the first cholesterol-induced mechanism caused by cholesterol. The second thing is that cholesterol inhibits the cholesterol synthesis. It's like a feedback regulation, So it's going to increase the intracellular level of cholesterol. It's going to activate the ACAT, which is an enzyme. The ACAT allows storage of cholesterol within cells. It's going to lead excess circulating cholesterol. So I want you to focus here. This is caused by high cholesterol level, and then it's going to inhibit the cholesterol synthesis. Okay, second thing, intracellularly, the level of cholesterol is going to be high. We have cholesterol high, which is going to activate the ACAT. The ACAT is going to store the cholesterol within the cells. This will lead to excess circulating cholesterol. So we have low LDL receptors, increased circulating cholesterol. This is the rule, leading to atherosclerosis. Because LDLs are basically derived from the LDL containing cholesterol, taking it to the walls of the vessels, 30% of it, at least. So LDL receptor endocytosis. So when the LDL, so this is the mechanism. It's called the LDL receptor endocytosis. So genetic deficiency of LDL receptors. If some patient had this LDL receptor deficiency, it's going to be always having an elevation of LDL in the blood. No uptake of the LDL is going to be in the blood always. And that will also cause elevation of the cholesterol. So this is another disease we have for today, the type 2 hyperlipidemia, familial hypercholesteremia. So we're done with the LDL, the low-density lipoprotein. We said that the low-density lipoprotein contains cholesterol derived from the VLDL and transferred to the extrahepatic tissue. The high-density lipoprotein, the healthy, the good one, actually comes to the vessel and takes up the cholesterol deposited by the LDL and takes it up and form the HDL to the liver. But how? We're going to see each step together. So the cholesterol taken up by the HDL, first we're going to have the nascent HDL, which is a discoid shape, as in disc. This is a disc. This high-density lipoprotein discoid shape is the nascent HDL, which is immature, initial stage. We have ApoA1, which is the structural apoprotein of the HDL, and ApoE and ApoC2, as I said in the HDL before. So ApoA1 is important on HDL. It's unique. Why? Because it activates the LCAT, the lecithin cholesterol acetyltransferase, which is present in the plasma. So focus on this picture. This is the cholesterol deposited by the LDL on the vessel. The HDL will come and take up these cholesterol, form the discoid shape, and the HDL actually has the ApoA1 that activates the LCAT. The LCAT is actually present in the plasma, just kind of roaming around. The LCAT is going to be activated by the ApoA1, and then it's going to bind when the LCAT is activated, it's going to make the cholesterol move, and it's called cholesterol efflux, move and goes into the HDL, and the HDL now is mature because the cholesterol moved from here to here. As you see, cholesterol moved, cholesterol efflux, caused by the LCAT enzyme, activated by the ApoA1. Now, the discoid HDL is actually a spherical shape HDL. And this is the mature HDL. Okay, so let's talk about it one more time. HDL is formed and how? First, the nascent HDL is a discoid shape. How did it become a mature one? Because it has the ApoA1 that activated the LCAT. The LCAT actually made the cholesterol move from here to the inside of the HDL, causing the HDL to be spherical, and because it contains now cholesterol inside of it. Got it? When ApoA1 activates the LCAT will bind to HDL disk and cause cholesterol particles to efflux to the HDL to make spherical HDL. Now the lecithin, so the LCAT is actually a lecithin, cholesterol acetyltransferase enzyme. Where is it present? So the LCAT is in the plasma, but the lecithin itself is a component of the phospholipid bilayer of the HDL. This is just important, was mentioned on the second carbon that's contained the poly unsaturated fatty acid, that's going to transfer the so this is the HDL, okay? The lecithin is just a part of this bilayer of the HDL, okay? So the lecithin in the purple is a part of the HDL on the outer side of the phospholipid bilayer, and it contains, the second carbon of this lecithin contains a polyunsaturated fatty acid. This is transferred to the third hydroxyl group of the cholesterol, and when this reaction happens, caused by the LCAT enzyme, when this reaction happens, it forms cholesterol esterase. It esterified the cholesterol inside the HDL. So now the cholesterol ester is more hydrophobic. It's going to move to the inferior of the HDL disk. Then this reaction will actually convert the nascent HDL discoid shape into a spherical HDL. So basically this is the mature HDL. Now it's spherical. It contains the cholesterol ester. Esterified because it's more hydrophobic than the normal cholesterol. This is the mature HDL. After it becomes spherical in shape, it will move to the liver, and then it will take up by the liver using the ApoA1 mediated receptors. Now, we can call it an anti-atherogenic and healthy cholesterol. It's very important. Using ApoA1 mediated receptors used in the liver to synthesize the bile acid. So, now we're going to talk about the last disease we have today, which is the Tangier disease, the disease of cholesterol transport. If this is caused by very low HDL, if we had very low HDL, and this is causing an enlarged liver and spleen caused by mutation, the transport protein, the cholesterol cannot properly exit the cell. So it's actually a very low HDL level causing the Tangier disease. The problem is with the cholesterol transport. The first case, so this is actually a very important disease because we have the significance here is very low HDL and gonna cause enlarged liver and spleen because of mutation in the transport protein of cholesterol, because not well transported cholesterol can cause it cannot properly exit the cell. It cannot bind to the ApoA and form HDL properly, so this results in low HDL, therefore enlarged liver and spleen. The last thing we have today, which is the high HDL, cancer, kidney disease, infectious disease, autoimmune disease, lung disease, and type 2 diabetes mellitus can cause high HDL, and mortality rates could go up. So that's it for today. I am so thankful for you watching, and thank you so much.